viewers, in this video lecture I will talk about a new term called Harmomegathy, a term used in pollen biology. Harmomegathy is a phenomena in which pollen grains exhibit characteristic folding along their apertures to avoid excessive loss of water and death. The term was coined way back in 1935 by Roger P. Woodhouse, a palynologist. Harmomegathy is a kind of physiological adaptation exhibited by the pollen grains. By this adaptation, it avoids non-viability of the pollen grain. Desiccation is essential for the pollen grain also, but to what extent this desiccation will occur, that is a crucial thing. So this folding, it controls the level of or degree of dehydration that is necessary. To some extent, this dehydration makes the pollen grain resistant to heat and other chemical substances. So when the pollen grains they hiss from the anther and they reach the stigma, during that time period it faces the exposure to environment. And hot environment can cause death of the pollen grains also. They might become very much desiccated. So a type of folding is necessary, and this folding has also a pattern. We will see that. So basically, it's a kind of a physiological adaptation, you can say, that Harmomegathy, it occurs in a particular manner and very highly controlled manner. When the pollen grain falls on the stigma, it again becomes rehydrated and viable. Viability is not lost. As you know, cryptobiotic condition, it's the situation that occurs in bacterial endospore or maybe in the cells of these seeds where dehydration or desiccation process is there. Everywhere where there is a spore formation or any kind of resistant body that needs dormancy, this dehydration is related with that. So, but how much dehydration, the degree of dehydration is very, very important, and that is being controlled by this phenomena Harmomegathy. Folding, as I said, that it is very much controlled or it occurs in a pattern. If you see the architecture of the pollen wall, you can correlate this Harmomegathy with this architecture. It's a kind of, you can say, biophysics in which the exsinus wall is thicker and tougher, and it is very difficult to fold the exsinus wall. So the folding occurs near the apertures, and apertures are the region that are exposed to the environment, and apertures only contain the intine. So intine it is unable to stop water loss. Exine is made up of sporopollenin, which is water resistant. So whatever water loss is taking place through the apertures. The diagram, the panel shown here is of different types of pollen grains. The first one, that is the monocolpate pollen grain. You can see the folding occurs along the furrow, along the single furrow. It's a one folding because it's monocolpate pollen grain. The second picture, that is the tricolpate pollen grain. In tricolpate pollen grain, we see three foldings in a longitudinal manner, just at the position where apertures are there. The margins of the apertures, they come close to each other and shut the mouth of the aperture. In case of porous apertures, the folding will occur, it occurs like a depressions. The margins of the pores become elevated, and it's kind of a sunken aperture, you can say. Sometimes the folding may occur a kind of called mirror buckling, that is an irreversible process, and that has to be avoided. So Harmomegathy avoids this mirror buckling, and it controls. That means it's a highly controlled phenomena, and there might be some genetic or regulation to this, I'm sure. So in this video lecture, it was up to this. Hope you are liking these videos, and if it is so, please press the like button, subscribe and share. Thank you very much for watching this video too.